I will talk about the evolution of C. elegans-microbe interactions. And I would like to start with briefly introducing what my group is interested in and what my group is studying. So we're generally interested in the evolution of host microbe interactions. For most of our work, we use C. elegans as our model host organism. And in this context, we study the evolution of C. elegans pathogen interactions. We're interested in C. elegans immunity and also C. elegans natural ecology. And I will touch on both of these topics just briefly. And more recently, we've started to invest more in studying the evolution of C. elegans microbiota interactions. Then another part of my group is working on bacterial antibiotic resistance evolution, and this is a topic that I will not touch today during my talk. So I think most of you know C. elegans. C. elegans has become one of the most intensively studied model organism in biological research. This is due to numerous advantages of C. elegans for research. It's a very versatile experimental model. It's relatively simple to manipulate this worm genetically. It's transparent, so we can actually observe phenotypes in vivo. You can freeze the worms, they survive it. It has a short generation time, etc. And C. elegans has been studied in diverse biological disciplines since the 1960s and 1970s, more or less in all of the areas, in all of the different biological areas. But here it is very interesting and fascinating to see that almost all C. elegans research that has been done and that currently is being done is performed without the natural microbiota of C. elegans. So when C. elegans is usually studied in the lab, it is without the microbiota. The gut is empty, even though we now know that the gut is usually full of bacteria. This has a lot to do with C. elegans having been established as a lab organism. And for a long time, it was actually not really known where C. elegans lives in nature. And this is one example where you do find C. elegans in nature, namely in rotting apples. And C. elegans is particularly common in autumn when you actually have these apples lying on the ground. Throughout the year, you can find C. elegans also on compost quite easily. And this is where we usually sample C. elegans throughout the year. Yeah, since not many groups have been interested in the natural ecology of C. elegans, we're only starting to understand what the natural interactors are. And this is a sketch of the current knowledge. So we're only starting to understand what kind of competitors are around, what kind of vectors may be used by C. elegans for transportation, what kind of predators there are, but then also what kind of microbes are interacting with C. elegans. And I'll be focusing particularly then on the commensals and mutualists that we currently know. And here it is particularly interesting to note that the very first study on the C. elegans microbiome was published in 2016, only in 2016. And it was actually work from 3 different groups and in 3 different papers that were published in 2016. Only one study really looked at naturally isolated worms, naturally isolated C. elegans, and that was our work that was primarily based on samples taken from Germany, but also together with colleagues from France. This is work, by the way, that was primarily done by Philip Dirksen. And since this time, we are sampling C. elegans every year and characterizing its microbiome. And this is work done by a postdoc in the group called Julia Jonke. From this work, we know that the microbiome is distinct from the substrate. So in C. elegans, we have the advantage that we can sample both the worm and the direct environment. That is illustrated in this graph here. The filled symbols refer to microbiomes from C. elegans and the open symbols from the substrate from which C. elegans was isolated. And if you use some kind of PCA plot, you see that these are usually very distinct. From this work, we also know that the C. elegans microbiome is only conserved at higher taxonomic levels. We usually do not find high conservation at the single species or strain level. So if we do a core microbiota analysis or an indicator species analysis, then it's very few species that are identified below 1% of the bacterial species that we find. So we think that the exact strains and taxa may not be important, but rather the functions that they could provide. And that here C. elegans may be selective, but on the other hand, it is also possible that it's actually the microbes who are in charge and determine who's interacting with the worm in the end. What we have done now to change a little bit or to bring a new perspective into the C. elegans research field is that we have now developed a so-called C. elegans microbiome resource by choosing 12 representative taxa, which we have characterized in detail for its interaction with C. elegans. We also obtained genome information, metabolic network information, colonization data, etc. And this is now a resource that we provide to the community, the C. elegans community. The 12 strains can be ordered from Synaptitis Genetics Resource Center. And we hope that they will now be used to characterize all the various gene functions and the various biological functions that have already been characterized in the past by the C. elegans research community. And this is indeed happening. So we know that there is now research going on with these isolates on understanding aging in C. elegans, various physiological traits, etc. So what we'd like to show you today is a brief overview of some of the work that we're doing. It will be more or less only a glimpse on a couple of different topics that we have become interested in. One of these topics is, of course, understanding whether there is a meaning to the microbes that we find in association with C. elegans. And to get a first understanding, we selected 77 isolates that are representative of the C. elegans microbiome that we usually find in nature. These 77 isolates are from 15 taxonomic groups. And we also try to characterize several isolates for certain genera that are usually common, like Pseudomonas or Corbactrum. And for these isolates, we obtain complete genome sequences and use these genome sequences to infer possible functions, metabolic functions that can be provided by these bacteria. And this is work that from my group was done by a previous PhD student or postdoc, Nancy Obeng, in collaboration with a group of Christoph Kaleta and his then PhD student, now postdoc Johannes Zimmermann, who are experts on metabolic network model analysis. And one thing we saw quite early is that there is a functional variation between and within microbiome taxa. That is illustrated here on the graph on the left, where we plotted pairwise metabolic identity to pairwise 16S RNA identity. And we usually find that the taxonomic identity is higher than the metabolic identity, meaning that for closely related microbiome isolates, we find quite significant differences in their metabolic competences. And that is also illustrated in the end by this tree here, where the colors refer to a certain taxonomic group of bacteria that we here characterize, and that are then linked according to their metabolic competences. And you then often see that certain colors, even though they do usually form clusters, but they sometimes are also spread, indicating that there's variation in these metabolic competences. One advantage we have with C. elegans is that there is a lot of information available on this nematode. And for that reason, we can also look at to what extent the bacteria that we characterize are actually providing essential nutrients for the worm. And indeed, they do. The metabolic pathways that are present in these microbes cover or are capable of producing all the essential nutrients, with one important exception, that is cholesterol apparently is not produced by these bacteria. What for us is then, of course, even more interesting is that we can use this genome data and correlate it with phenotypic data to see whether there are certain metabolic units, metabolic components that are important for the interaction with C. elegans. And this is something I try to illustrate here. So here you have the codes for different bacterial isolates. The identical colors refer to the same taxonomic group of bacteria. And then in a green heat map, you see their effect on C. elegans when they are combined with C. elegans in monocolonization experiments. So here you see, for example, some variation among these strains in their ability to support population growth. Or you also see variation in the ability of these bacteria to colonize the nematodes. We then looked for an overlap of these types of heat map with the presence of certain metabolic functions, and indeed we found some. So trans-3-hydroxyalproline degradation is significantly correlated with the ability of these bacteria to induce population growth in the worms, and pyruvate fermentation to acetone is correlated with the ability of the bacteria to colonize the worm. These are obviously only correlations, and this is one of the challenges now to actually validate and prove whether these functions are really important for these phenotypic traits. We then also became very interested in immune protection by these bacteria. This is actually a topic that we cover for a very long time in the group. We are very much interested in C. elegans immunity. We noticed very early on that there are certain microbiome members that seem to provide immune protection against pathogenic bacteria. And this has then been studied more intensively by Katja Dierking and her subgroup. Katja Dierking is a staff scientist in my group, and she has her own subgroup, and it's particularly the work of two PhD students, Koa Kesoyan and Lena Peters. And they looked at isolates from a couple of different genera, Acinetobacter, Ochrobacter, Pseudomonas, that are listed here, and then their effect on worm population growth in the presence of a pathogenic bacterium, the pathogen Bacillus thuringiensis. And you can see that if you have certain Pseudomonas bacteria present, then the worms are not so much affected by pathogenic Bacillus thuringiensis. And this is similarly seen in a survival experiment. Here we have different concentrations of Bacillus thuringiensis, and here we look at the survival of worms in the presence of these different bacteria, here indicated again by their codes and the different color schemes. And here we focused already on the Pseudomonas isolates, and if these Pseudomonas isolates are present, then the pathogenic Bacillus thuringiensis do not have a strong killing effect anymore. The question is, how do they achieve that? One hypothesis is that the Pseudomonas isolates produce an antibiotic compound that kills pathogenic Bacillus thuringiensis. And this is something Koha tested with the help of a zone inhibition assay. And for two Pseudomonas isolates, MIP11 and MIP12, she could indeed prove that these bacteria produce an antibiotic compound that is effective against Bacillus thuringiensis. That is also shown here quantitatively for these two isolates. And here you have a positive control highlighting how large the zone of inhibition could be. Interestingly, none of these other isolates produced such a zone of inhibition, suggesting that they provide protection by a different means. Katja Dierking and her subgroup, they then joined forces with a biochemist, Helge Bode, and he identified the substance that was produced by these protective bacteria, and it is a substance that is called macetolide E, and it is indeed this substance that is responsible for the protective effect of these two isolates. So with the help of these experiments and these analyses, Katja Dierking and her subgroup, they could show that these two bacteria provide direct protection against pathogenic bacteria by producing an antibiotic compound. But there are other microbiome members, like the Pseudomonas strain MIP115. They provide some kind of other protection, some kind of indirect protection, as illustrated here. And this is something that is currently under study to better understand what other kind of protective mechanisms may be possible. Just for the two last slides, I will now switch gears because we are also very interested in understanding the bacterial side. And this is now work done by former PhD student Nancy Obeng and a current PhD student Anna Czerwinski. And here we wanted to better understand how bacteria adapt to a host system. And here we took advantage of C. elegans being a fantastic lab organism. We performed evolution experiments, something we have established over the last decade for this system. And we used evolution experiments to evolve, adapt bacteria to a host lifestyle. And we then combined that with phenotyping genomics and also functional genetic analysis. And I will only give you a very brief illustration of the results that we obtained, a very comprehensive data set that we collated over the last years. This is a sketch of the life cycle to which we adapted the bacteria. So we assume that these bacteria often have an ancestral free-living lifestyle. And we now imposed a life cycle consisting of two phases in the free-living environment and then in host association. And we believe that this kind of biphasic life cycle imposes strong selection at several steps, at several different stages. The bacteria need to enter the host, they need to be maintained in the host, and they also need to get out again and then proliferate in the environment again. And we adapted these bacteria, in this case, the Pseudomonas isolate, Pseudomonas lurida isolate, to this biphasic life cycle, and then tried to understand where adaptation took place and what kind of genetic changes are responsible for these adaptations. What we found out is that the dynamics are driven by bacterial adaptation to host entry and maintenance, so particular to these first two steps. And we could demonstrate that the bacteria that we experimentally evolved indeed improved their ability to enter C. elegans and be maintained in C. elegans. And the genomic and subsequent functional genetic analysis additionally indicated that genetic changes in genes like WISP-E and WISP-F are very important here. And this is something that we're currently characterizing in more detail in the moment, and I'll be hopefully telling you more about this in the future. With this, I'm at the end of this short overview. C. elegans, in my opinion, is a fantastic system to study the evolution of symbiosis, particularly as we can perform evolution experiments to understand the evolutionary dynamics of these kind of interactions. And we can combine it with functional genetic analysis of both host and the microbes to understand the underlying genetics of these kind of interactions. With this, I'd like to acknowledge financial support by a variety of sources, but particularly of the Collaborative Research Center, Origin and Function of Meta-Organisms, which I already introduced to you earlier. I also like to acknowledge a couple of colleagues with whom we collaborate in a very constructive and successful way. And I'd like to thank you for your attention.
Thank you very much, Hinrich. We have time for a quick question. Are there any, if you want? Yeah, Sudeep, please.
Hello, hi. Yeah, very cool work on C. elegans. I'm interested in that side which you probably didn't mention that much, but the non-bacterial dependent immune protection, where you see it's not due to bacterial competition or production of any antibacterial product, but rather a different mechanism. So what do you think is happening? Is it immune priming, or is there any kind of immune priming present in C. elegans which would tend off other incoming pathogens?
That's a very good point, and it is research in progress. So we were testing the idea that these bacteria indeed activate the immune system. There are some indications that this may indeed happen.
Okay.
From the work of other groups, it is known that some kind of immune priming is possible. So far, we did not find it elicited by the bacteria that we are using. So at the moment, I cannot really answer your question, except that there seemed to be an activation or an activation of the immune response is in principle possible by these bacteria. We do not know yet whether this is really the important factor here.
May I ask in follow-up, what do you use to check? Like, do you use NF-kappa-B activation as a measure, or of immune?
NF-kappa-B activation is unfortunately not possible because the worm does not have that. But we have a variety of, so there are different ways how we can do that. So either we do this by transcriptomics, it's something we have already done in the past. But since the C. elegans immune system has been relatively well characterized, there are fluorescent reporter strains available. So where fluorescent protein is linked to the promoter of known immunity response genes. So we can use that to look at the activation of different immunity response pathways. And this is indeed something that Katja and her group are currently doing.